Good. Well, first of all, a very warm thank you for the honour and privilege of being here today to celebrate your 99th birthday and the beginning of your 100th year. It really is quite an enormous achievement. Also, just this week to be ranked #1 again in the university rankings, so that's wonderful news. Now, what I'm going to do today is to talk about pandemics. I'm going to give you some slightly unconventional views about what are the real risks coming in the future, and in a sense turn to the problem of how international agencies and governments can address some of these risks. So if we start with the origins of human infections, these things are fairly obvious. We inherited most from our ancestors, and I'll come to our ancestors in a minute. We acquired a great number from wildlife, and that has been particularly true in the last 100 years or so. And then, increasingly, we acquire quite a lot from livestock. These are more silent ones. They tend to be bacterial in nature. If one takes the total burden of infections from viruses through to protozoa and insects, then about 60 to 70% of these are zoonotic. In other words, the human host is only one part of the life cycle, and often the maintenance of the infection, such as Ebola, is maintained in other mammalian hosts. And that's a very, very important point in terms of your efforts in the Netherlands, and at this university in particular, to look at One Health, which combines a veterinary and a medical approach. If we turn to ourselves, our time on this earth has been quite recent. If we take our subspecies, Homo sapiens sapiens, we are somewhere of the order of 100,000 to 150,000 years old. If we take Homo sapiens as the species, then we're going back a little bit further. Now, as a sort of population biologist, epidemiologist, what I translate that into is the number of generations of humans that have been on the earth. And a generation is defined as the time from the birth of a female to when that woman provides the birth of another female who reaches sexual maturity. In other words, to perpetuate the demography. And if you look at our subspecies, then we have had about 10,000 generations. So the opportunities for evolution really are quite limited. Now, if I then turn to influenza A, and now over the same time period of about 100 to 150,000 years, ask how many generations has influenza A had, then clearly that is trillions. And so the pace of infectious agent evolution is infinitely faster than our own. Now, how do we deal with that in modern society? We deal with it with medicine, modern medicine. But as many of you will know, from point of discovery to market, for a small molecule drug is about 10 years at average at the moment. For a vaccine, it's about 8 years. So although we've speeded these processes up, we're still well behind the evolutionary rate of the agents that we're trying to confront. Now, if one looks at what we've solved and what we haven't solved, this spectrum of genetic variation is very revealing. So if you take the relatively homogeneous end, we have effective vaccines against all these agents, and then we go through to the right end, HIV, despite intensive research over what is now a depressingly long period, 30 years, we have no interesting HIV candidate vaccine. And similarly with malaria, we may see in 2017 the licensing of a new malaria vaccine with an efficacy perhaps in the 60 to 80% domain, but that has been approved by WHO for the use in 3 countries in Africa, and we will see what happens. And the reasons are trivial. HIV is a moving target all the time. Every single evolutionary change that can occur by substitution, deletion, etc., occurs every day in an infected patient with a high viremia. If we take malaria, it's even cleverer because it has 3 mechanisms of generating variability. 1, mutation through asexual reproduction. 2, most importantly, sex, recombination. But 3, even more importantly, it can switch on and off copies of the gene which are slightly different, which code for surface antigens. And so malaria is a super genetic variant. It changes too all the time. And so at the left side of this, we've solved these problems. At the right side, we're still struggling scientifically. Now I want to turn to the topics that really are of greatest relevance to One Health, or Global One Health, and it is extraordinary that our modern basket of technologies allows you, of course, to do things that even 5 years ago were not possible. And one is whole genome sequencing, which is looking at the genetic code of a pathogen and looking at its close associates in animal or other communities, and particularly primate or cattle, livestock communities, and ask questions about hybridization. In other words, do human pathogens actually mix and hybridize with cattle and other pathogens in our livestock? And the example I just wanted to share with you very quickly is quite a remarkable one because it's just come out this year, and it's to do with a very common disease. I should just say that these chicken and livestock densities on a global scale are very important. They march roughly in tune with population density, but not quite. So you can see that Europe is very, very heavily populated, and as you know, the Netherlands has the highest density of chickens per person per square kilometre. And when you ask students about that, when they start studying, they always say China, but it's not, it's the Netherlands. Now, these densities are very important because they're opportunities for evolution and transfer. Now, the example I wanted to mention to you, which was a recent paper, followed up from this paper, which was the beginnings. And essentially, it looked at hybridization of schistosomes, which cause bilharzia, a very serious disease of humans. And this is schistosoma hematobium, which causes quite a lot of damage around the bladder and can actually cause mortality in young children through secondary bacterial infection. And this study by Bonnie Webster and colleagues showed that as cattle, the bovine schistosomes, were commonly hybridizing with the human form, and these hybridizations were fertile, and when you took them into animal models, they produced viable offspring. And what it tells you is that evolution is continuing. And as we have this intimacy with humans and livestock, many more of these events, even in these big genome organisms, which are big, sophisticated genomes, these hybridizations are going to become more and more important. It doesn't need me to say that antibiotic resistance obviously is probably the premier example of this, and the flow between us and livestock and coming back from livestock to us in terms of antibiotic resistance, perhaps captured by plasmids, is very frequent. The interesting thing is the techniques to map this transfer are now becoming, as it were, platform technologies which permit us to look at this gene flow across the pathogens of livestock coming into humans. And in this university here, you have special expertise in that domain, as you do across the Netherlands. Now, I wanted to, just before moving to pandemic control to end on, I wanted to concentrate on what is changing in our world which influences these things, that the spread of organisms, both between livestock and ourselves, between wild animals and ourselves, but most importantly between ourselves and ourselves in different continents. And the most obvious, of course, is that population density is growing to 9 billion by 2050. Nothing is going to stop that. It is extraordinary. When I was born, the population of the world was about 2.5 billion, and we're now up at 7 billion. It is the most extraordinary change. This is important for two reasons. One, higher population density, more frequent transmission. But even more importantly, every transmission event is an opportunity for evolution. And therefore, higher human population density promotes transmission, but it also promotes evolution of the organism, the pathogen. Perhaps most importantly is this slide. This comes from a different discipline, geography. And geographers tend to chart lifetime tracks of people. And the four families here from Britain, great-grandfather from the East Coast, grandfather coming forward to father. Now we start with the great-grandfather. He walked between villages, perhaps got on a horse sometimes. His son, the grandfather, went by coach and horses between counties. And his son, which was the Second World War generation, and you can see that in the footprint of his travels, plus post the war, a little bit of holiday travel. And then my generation, the son of the Second World War generation, we're totally global trotters, as it were, across the world. And the current generation, the young generation, the university students, are equally globetrotters. So out of our 10,000 generations as a species, within 4 generations, we've turned from a local domestic animal into a globe-trotting species, and that has enormous implications for the spread of pathogens. This slide, sorry, my apologies. Can we come back to that one? Yeah, the red one. It's just setting this one running. Yes, there we are, thank you. At any one time, the average figure for the moment of the number of us up there in the aeroplanes is about 650,000 people at any one instant in time, bringing all their bacteria and viruses across the world. These are the flight patterns. This database is held in Los Alamos. As an academic, you can get access to it. And it's got a 24-hour moving clock on it. It's daylight in Europe at the moment. And then this moves across to North America, and you can see the change in the flight patterns. Every yellow dot is an aeroplane. The most dramatic, if you take the time series of this sequence, and you go back to the 1980s, then you see the dramatic change has been in Asia, the connectedness of China. And this essentially dictates how infectious agents spread throughout the world, and I'll give you an example in a minute. This slide was produced for Margaret Chan as Director General of WHO, and it's 18 months compressed into a few seconds. It's influenza A evolving in Cambodia, and through the linkages of air travel and people movements, it shows that within 18 months, the green zones are infected or dead, and the epidemic of influenza A will spread globally within a very, very short period of time. There's absolutely nothing we can do about that. The French in the H1N1 epidemic had the good idea of restricting travel between infected areas and France. You can do a calculation which tells you instantly that that's a waste of time. By the time you detected it and acted, the virus is already in your country. And whole genome sequencing of the influenza virus, the H1N1 virus, showed the transmission of this from rural Mexico into Mexico City, and then within literally 5 weeks, that virus was spreading internationally via the global airline network. What else has changed? We're becoming more urbanized. The number of megacities, which are cities over 10 million, has grown enormously, particularly in Asia. And this brings the intimacy of the contact between livestock and humans, because the peri-urban areas are where the livestock are brought into. This is an example of Hong Kong, which is a particular sentinel site for monitoring influenza. So that has changed. And then, of course, much of this has triggered a series of recent events, which I'm only going to mention 3. First of all, we have as a society an obsession with safety, which spills over into the public interpretation of scientific advances, and vaccines are probably the most important innovation in medicine, which has contributed most to the reduction of child mortality. And yet we're in a society at the moment where increasing concerns are expressed in all sorts of quarters about vaccination. Fourthly, in terms of the epidemiological analysis of the data, this is one example from Britain, where Andrew Wakefield's paper in The Lancet, which was subsequently disproved and thoroughly discredited, led to a very significant drop in the uptake of measles vaccine, and that has triggered epidemics in the recent years. And I've seen similar things in the Netherlands, and I've seen similar things recently in California. The second thing is an increasing attention on health economics, which is taking the spread of infections, whether from animals to humans or human to human, and calculating that if you control it by vaccination, what is the benefit to the health system of that society? And one of the most surprising results to me recently was influenza A, that in a sense, this is a very good paper by a very good group, which showed that influenza A vaccination for all age group is health beneficial. And therefore, in a sense, it's not just the elderly and the young, it's the herd immunity created that will stop influenza transmission. Now, Ebola, we know the problem here, and it's a very sad epidemic in many senses. We all were horrified at the pictures of the mortality that was occurring in families in the west coast of Africa. But to epidemiologists, Ebola was never a threat on a pandemic global scale. for a few very, very simple reasons to do with its transmissibility, the onset of clinical symptoms, first before highly infectious state, so isolation and quarantine could work very well in controlling that epidemic. So as sad as this was, it wasn't something that was going to create a pandemic, despite what was said in the press and indeed by some very senior people in America. If we turn to the outcome of this, probably the saddest bit, and it's going to be where I end, is on vaccine development. Because vaccines were developed, but as the cases started to fall, the clinical trials designed to test the efficacy of the vaccine fell below the number of cases required to test the vaccinated versus the unvaccinated in terms of the rate of accrual of new cases. So all these phase 3 trials were stopped. Zika has been much in everybody's minds, and a vaccine through Sanofi Pasteur looks possible but problematic. Zika is going to become endemic worldwide, there's no doubt about that. It will be an oscillatory, rather like dengue. There will be bad years and good years, but this virus, this strain of this virus, will become endemic. And indeed, the pathology, particularly microcephaly in children, is beginning to become a noticeable morbidity event in America, US. And that is, in one sense, of course, very, very sad, but in one sense it's good, because it will stimulate the development of the vaccine. Now, lastly, I just want to move to SARS. This was another Ebola-like problem. To epidemiologists, SARS was never going to become a global pandemic, for a very good reason. The onset of clinical symptoms were well before peak infectiousness, and if you isolated a patient before, just after the onset of the clinical symptoms, which are high temperature, then you could stop transmission dead. And that's exactly what happened with SARS. But do note, as with Ebola, this is another bat origin virus, and it is still an ecological puzzle why bats harbor so many viruses which have this ability to transmit to humans. And clearly one major thrust of scientific research internationally at the moment is to understand a lot more about bat viruses so we can somehow turn to predicting what might infect humans in the future. The clinical side of SARS and influenza A is illustrated here, and this shows why we always will be able to control a SARS-like virus, but we will never be able to control, by public health measures alone, an influenza virus. If you look on the right-hand side, you see post-infection, these are medical students, volunteers, 2 days after infection, peak viremia, peak symptom score, peak infectiousness. With SARS, you can see that post-infection, it takes 17 to 20 days to get to peak infectiousness, and symptoms occur at day 5. So you've got 15 days to get that patient isolated before he or she spreads on to others. With influenza A, by the time you and I, if we were feeling seriously ill with influenza, went to a physician, we'd already have spread the virus on to others in our family and our work colleagues. I'm going to jump now because of time, and I'm going to go straight to control of pandemics to end. Now, many people feel that the control of pandemics lies largely with generating either a small molecule drug treatment, an antiviral or an antibacterial, or indeed a vaccine. But I would put this the other way round. Most of these, the platform technologies for developing the vaccines are there. The Ebola candidate vaccines were up within about 6 weeks from the epidemic starting. What took a long time was phase 1, 2, and 3 trials, safety in humans, going into a field situation to test, and those trials take a lot of time and a lot of patients to do. Now, if you're a pharmaceutical company who has the platform technology, why would you develop a vaccine, and this is a harsh commercial reality, when the vaccine doses you're going to produce are about 1 million perhaps? What is the financial model that will take that vaccine into the store afterwards? And MERS is the same. Zika is slightly different because I think endemically worldwide, that will be a major problem, and there will be a commercial opportunity for companies. And so my argument is that the problem with vaccine and solution to pandemics is not science and technology platforms, nor is it the ability to do clinical trials. It is developing a financial model internationally which would fund these things. And as some of you know, a new organization has just been established, CEPI, which, not quite sure where it's going to be based, although it's starting in Norway, which will attempt to create a financial model which would fund the vaccines for these emergency outbreaks. At the moment, it's a twinkle in a lot of people's eyes, including Bill Gates, who has very kindly offered to fund a non-trivial part of this, but I still worry that nobody's come up with a proper financial model to turn from the trial of the vaccine into its large-scale manufacturing. We have deep problems with dealing with fast timescale epidemics, and an epidemic characteristic, as illustrated here, is controlled by two parameters. One, a basic reproductive number, which is number of secondary cases generated by one primary case. For influenza A, that's about 1.5. But the most important parameter determining this shape is the generation time, which is the time from a first infection to when a person transmits onto another. And influenza A is the fastest we know, it's 2 days. And that determines the doubling time of the epidemic, so you're always going to be behind the progress and the spread of this organism. So if you look at international worry lists, unfortunately in Europe at the moment all our governments have terrorism at the top, but if you go down off terrorism and look at the next level, it is influenza A. It's still regarded as the major international threat. The oddity is the tasks here we can do very easily now. Unusual clusters of morbidity and mortality, the internet and reporting, Facebook, etc., provide an extraordinary global sentinel of events. Identifying the etiological agent is almost standard now, with looking for unusual RNA and DNA. Developing a diagnostic test can be done in a few days. Determining the route of transmission takes a little bit longer. It requires epidemiological studies in families and groups of people. Developing the algorithms for clinical care is often very problematic because you can't do a clinical trial. You're working on hoof, as it were, trying rapidly to work out what does work and what doesn't, and then putting in place the activation capture of the data to see how you're doing, and of course in all of this keeping the public informed and identifying and implementing key policies is central. I want to end just on one of the issues about helping politicians identify the key policies. Netherlands, like the United Kingdom, like every country in Western Europe, has a document like this. It's called Pandemic Planning. And I suspect your document's rather like ours. It's very weighty. It's constructed by vast numbers of committees from very informed people, and it has 101 options that supposedly cover every eventuality. Here's the crunch. What is your primary policy objective? Is it to minimize morbidity and mortality? Do you have a fixed or variable budget in your Department of Health to do that? Do you want to buy as much time as possible so you can develop a vaccine and get it manufactured to put it into place? Do you want to minimize the duration of the epidemic so you minimize the disruption to the economy, particularly if you're going to close schools? Or do you want to minimize the peak prevalence because that could saturate your tertiary care, the hospital care, such that you would not be able to cope with the patients, as occurred in the 1912 pandemic of influenza? So you're sitting opposite the Minister of Health and you're saying to him or her, Minister, which of these would you like to do? And you know what the answer is, all of them, please. Now, scientifically, you can work out you cannot do them all at the same time. This is a game theoretic problem. And therefore, you have to then go back and say, which is your top priority? And that usually causes a lot of problems. This is the ranking that was produced in discussions in the UK during the H1N1 epidemic. I think personally as a scientist it's the correct one. your primary task is to minimize mortality. Thereafter, there are a series of economic issues, but it does illustrate the problems of science here, that in trying to control an epidemic, your options are quite limited and some are mutually exclusive. Now to end, this is the salutary bit of us living in the Western world. Inside the circle, there are more people living than outside the circle of our world. If I look at world vaccine manufacturing technology capability and manufacturing capability, it all lies in Western Europe and North America. There are glimmers, certainly in India, and there are hopeful glimmers in China, but not quite yet. So if a pandemic emerged and we changed the regulatory structure for taking things through clinical trials, so we could probably do it in 2 months, platform technology, clinical trials, safety, into humans, then we would be all right in Western Europe, North America would be all right, the rest of the world would have deep problems. And that is something in this notion of global One Health that we really have to start to address. The number of vaccine manufacturing companies in the world has reduced from something like 8 serious players to at the moment only 3. And it may be in the coming few years that number will reduce again. But our task actually in the rich world is to try and encourage, not a commercial competitive consideration, but to encourage the manufacturing capability for these countries in the poorer regions with technology aspirations to actually develop manufacturing for vaccines. So lastly, talking more to the students than my own age group here, in your lifetimes you will see the emergence of many more novel infectious agents. And that emergence will be more frequent in the coming decades for 2 reasons. One, of course, we're better able to detect something new, but more importantly is our population expands, we travel more, and we encroach on natural and wildlife habits, and we need to produce more livestock to actually feed this growing population. And the intimacy of our relationship there in the global One Health will be an issue in terms of these transference. Modern medicine can solve many of these problems now, whether it's small molecule or vaccine, but the regulatory structure in place that we have mitigates against rapid action. And WHO struggled with this during Ebola, and it required a very clever academic, somebody may know this individual, Mark Lipschitz at Harvard, to suggest to WHO that they throw out the window randomized, controlled, placebo-controlled clinical trials and do something totally different in an emergency. And when he first made his suggestion, it was just thrown out of the window by the authorities, but today it's very much back on the board. So the regulatory structure and the manufacturing need to alter. Developing the technology for the vaccine is trivial now, it can be done instantly. And One Health, just to come back to your goals, and I've said this before to Professor Moll, that the Netherlands is in a very unique position here, because you in Europe have led the way in many ideas concerning the control of antibiotic resistance, the reduction of the use of antibiotics in the veterinary practices. You have a very sophisticated and high-quality medical university structure here, and you've taken this initiative of trying to draw these 2 communities closer together and, of course, linked to agriculture. And I should end on the point that do note that mortality from most of the common childhood infectious agents fell dramatically well before vaccination started in the mid-60s. And the reason for that was simply nutrition, because vaccines only work in a well-nourished child. And so nutrition, the veterinary transition, humans and back, and human medicine, you're in the Netherlands in a wonderful position to take a world lead in this domain. And I just end, remember, influenza A is the biggest threat. Keep having your annual vaccination. Thank you very much for your attention.